So now that we know kind of the basic types of cells, we need to look at what a cell is composed of. First off, we're going to talk about the cell membranes. And the cell membrane or plasma membrane surrounds the outside of the cell, and when we get into the organelles, it's also the same type of membrane that surrounds each organelle. It has a relatively simple structure. It's composed of phospholipids, and phospholipids have two different properties. At one end of the molecule, they have a hydrophilic head or a polar end. And hydrophilic, hydro means water, and philic means to like or to love. So this end of the molecule likes or loves water. The opposite end consists of hydro again, hydrophobic tails. So this end of the molecule does not want to associate with water. So it's got a polar head and that non-polar tail. So if you throw a bunch of these phospholipids into water, they'll spontaneously form a sphere structure. They'll tuck all of the tails to the inside and squeeze water out of that region. All of the heads will be in a layer on the inside with water trapped, and also on a layer on the outside with water excluded on the outside to form a big hollow sphere. And that's the property that really holds all of your cells together, is this phospholipid bilayer. And we'll see that it has lots of different functions and lots of different proteins and things that allow molecules to get in and out of the cell around this phospholipid bilayer. So it's a fluid mosaic model, and explaining the fluid mosaic model, the best description I can come up with is if you think of a swimming pool filled with ping pong balls. So if you take a swimming pool and fill it with ping pong balls and you look across a surface, it'd be like if you were able to stand and look across the surface of your cell. It would look like a uniform surface of all of those ping pong balls floating. Well, each of those balls is not attached to the one next to it. They're able to freely float. The thing that the swimming pool analogy can't really show is that there's two separate layers. So here's our top layer of our ping pong balls with their tails hanging down, and then this second layer, which is that bilayer, the second portion of it, with again the balls to the water side and their tails hanging up in this diagram. So because each of these phospholipids is not connected to the next, they can freely move around, which is why it's a fluid model. Also, any proteins embedded, we can see we've got some transmembrane proteins in there, some surface proteins. Those transmembrane proteins are also relatively free to float around. So it'd be sort of like if you had your swimming pool filled with ping pong balls and you throw a couple of soccer balls in there, or a volleyball. Each of those can freely float around as well in between all of the ping pong balls. We'll talk about the specific membrane molecules. We've got transmembrane ones that pass completely through both layers. There are surface proteins, which are only attached to the surface, either on the inside or outside of the cell. Oftentimes, those surface proteins have carbohydrates or sugars hanging off of them, and we'll look at their importance. Also, one more molecule that's essential for your plasma membrane is cholesterol. And cholesterol, actually, because it's a fatty molecule, will fit into the inside of that bilayer. See, there's several cholesterol molecules here. And what those do is they tie together the tails of a couple of different phospholipids, kind of their immediate area phospholipids, and that helps give strength and stability to that plasma membrane. Be sort of the equivalent if we took our pool of ping pong balls and we strung together little patches. So maybe we string together, just by running string through the ping pong balls, patches of 20 or so at a time around the pool. It gives a little more structure and stability to your phospholipid bilayer. So cholesterol is important, but not too much of it. So our types of membrane proteins that we'll look at. First one is a receptor protein, and receptor proteins oftentimes receive hormonal signals from the cell. So here's our kind of generic hormone. It fits into an active site on this transmembrane protein, and it does not actually pass through. The protein itself, this large transmembrane protein, just relays the message. So we have a hormone attached out here, and it sends the message to the inside of the cell without anything actually entering the cell. We have lots of recognition proteins, so oftentimes those recognition ones are the ones with the carbohydrates hanging to the outside, and we'll look at some specific examples of that throughout the class. Your blood type, either A, B, AB, or O, are all examples of the different recognition carbohydrates or sugars attached to your red blood cells. Membrane proteins can work as transport to bring molecules into and out of the cell, and in that case this is just a hollow tube. Sometimes they require energy, other times it's just passive. Anything that fits through will go through. And some membrane proteins are enzymatic, so they'll carry out reactions either on the inside or outside of the cell. Cell membranes are involved in quite a few diseases. We'll look at just a couple of them here in class today. First of all, cystic fibrosis. Cystic fibrosis, they have a thick or sticky mucus that's produced in the lungs, is what we primarily think of, but also throughout the entire body, all the other internal organs. And that thick, sticky mucus is the result of one single transmembrane protein that's not functioning properly, not moving stuff in and out of the cell the way it's supposed to. And so that causes the symptoms of cystic fibrosis. So you have to use kind of very firm beating on people's backs to loosen up that mucus so they can breathe properly. Another, not really a disease, but very uncomfortable, another symptom that comes with membrane proteins is anxiety. So some people have very intense anxiety in certain situations. Looks like this gentleman here is standing up to give a speech, and for people who are very afraid of that, they produce adrenaline. And that adrenaline will bind to receptors throughout the body and trigger different physiological symptoms. So they'll start to sweat, they will start shaking, their heartbeat will speed up and become very rapid. So all of those are the effects of adrenaline that's released. Well, if people know that they have these anxiety issues, they can take a medication called a beta blocker, and the medicine has the same shape as adrenaline, but it doesn't pass the message. So before they give a speech, they can take some beta blockers, and they sit right down in those cell receptors and prevent the adrenaline, the real message, from getting through. So that means that they can give their speech and they don't shake or sweat profusely or have a rapid heartbeat because they've blocked those membrane receptors. Another important thing, and I've already mentioned this with our ABO blood groups, is cell identification. So your immune system uses this to recognize if a cell belongs or if it doesn't. This is why when we have organ transplants, they're matched very carefully from the donor to the recipient to try to get the best match. And even with a really good match between the donor and recipient, it may not be exact. So this has kind of a purple and a green molecular marker. Well, the recipient here has two purples. So to prevent their body from rejecting and attacking that organ, people who receive organ transplants have to remain on medication for the duration of their life to suppress their own immune system so it doesn't attack that new organ. This is also why you're careful about which type of blood you give in a transfusion, because if you give the wrong type, your body's just going to attack it. These identifying markers are also used by things like HIV. So HIV is an infectious agent, and later on this semester we'll talk in detail about the HIV infection cycle. But it actually recognizes and binds to receptors on specific types of cells, these CD4 markers, and so HIV attacks the correct type of cell for the infection in your body. We'll look at types of transport, and two main types are passive and active. We'll look at passive on this slide. So passive transport does not require any energy. It could just be random diffusion, so if molecules are small enough, so very small molecules, they can slip right through that phospholipid bilayer. Remember, those are all just ping pong balls floating, so something smaller than that could fall right through. Anything that's larger will require a channel. So there are lots of passive channels, so this is just an open protein tunnel that goes through that phospholipid bilayer, and any molecules that are small enough to fit through the tunnel can go through. Usually they're fairly specific, but anything that fits will pass through. Simple diffusion is where it's just randomly crossing. Facilitated diffusion requires that protein channel, so it's facilitated or enabled by that protein channel. Osmosis refers to water. Osmosis is specific to water. The movement of water across the membrane is osmosis. A good example of a passive simple diffusion movement is in this cup of water here. If you add a drop of dye, the molecules of dye are going to be very tightly packed together when you first put it in. Gradually over time, though, those molecules are going to spread away from each other until they're evenly distributed throughout our cup of water. So a little bit more on osmosis. We mentioned it on the last slide, so we know it's diffusion of water across a membrane. And several different types of solutions. One would be an isotonic solution, and that's where it's balanced. So you have the same amount of water going into the cell as going out of the cell. And the important thing here are the things dissolved in the water. So notice there's about the same number of kind of random red spots outside as inside each of these cells. And those represent different things dissolved in the water. If it's a hypotonic solution, what happens is you have higher concentrations built up inside the cell, so the water is going to try to get inside the cell. Basically, it's an attempt to dilute what's inside the cell, so it's spread out the same as what's on the outside. Kind of nice space between each of these molecules. However, in animal cells, we don't have any strong structures on the outside. So in animal cells, if we have too much water, so this is a red blood cell, say in our bloodstream, the water rushes into the cell and the cell will burst open. And this actually has caused several people, or actually lots of people, to be hospitalized because they take water in too quickly. So marathon runners, a very bad idea for like fraternity and sorority hazing, drinking large volumes of water in a short period of time. And some people have actually died from drinking large volumes of water. This is one of the reasons, because they develop this hypotonic solution in their bloodstream and they start destroying their red blood cells. Plants don't have that problem because they have a very tough wall on the outside that holds them together. Opposite situation, hypertonic, is where you have more things on the outside dissolved, and so it's going to pull water out of the cell. So it's going to dehydrate the cells. And you can do, we probably won't have a chance to do this in lab, but very easy experiment to do at home is to test this using celery in water, either just distilled pure water. The celery will perk right up. Throw some salt in the water. So now we have lots of molecules outside of the celery. The water's going to rush out of the celery and it will fall over and get very limp. And this doesn't take very long either. So try it at home and your celery will keel over and lose water very, very quickly. So the other type of transport is active transport. And active transport moves molecules against the concentration gradient. And if we compare that to our passive transport, our molecules are moving down the concentration gradient, so from highly concentrated up here to nicely spread out. Well, our active transport is the opposite. So we're taking them from a spread out area and forcing them to concentrate more. So it's going against the concentration gradient, and this is why it requires energy. Your cells use energy of ATP, and one example of kind of transferring molecules to an area where they're already concentrated is in your stomach. So already your stomach is at an acidic condition. When you start to eat, that triggers certain receptors in the lining of your stomach to push more ions in there to increase the acidity of the stomach, so that as the food comes down, the acid isn't diluted out. So it keeps packing the acid into your stomach to handle all the food that's coming in. Moving very large things either into or out of the cell requires either endocytosis to bring large molecules in, or exocytosis to get large molecules out. And the cartoon version of it is down here. If we have something inside the cell that we want to get rid of, we're going to use exocytosis. Those molecules will be packed in membrane that will be pushed to the edge of the cell, and the membrane around the molecules and the cell membrane will fuse and open up and just basically spit out all of those large molecules. To move something into the cell, it's that process in reverse. The outside membrane starts to curve in and wrap around those large particles you need to bring in. Eventually these sides here will come together and seal into, it's called a vesicle, or just basically a little bubble of membrane to pull that substance into the cell. Once it's in the cell, you can do a variety of things with it, oftentimes just breaking open that membrane so that the cell can get at it. This is actually showing, this is a cell on a transmission electron microscope that's engulfing another large, almost the same size as itself, another large organelle. There's also receptor-mediated endocytosis, where specific receptors on the cell surface and on the molecule to be engulfed will communicate. So this happens to make sure that your low-density lipoprotein goes to the correct location. And so there are receptors on your liver cells that will recognize and bind to these LDL particles and pull them in. And then it can get at the lipids inside of there and make things like estrogen and testosterone. Bad part of that, if you have too much of that LDL circulating in your system, it can actually cause heart disease. So it can block arteries. A couple other things about cells. There are specific connections between different types of cells. The first one is tight junctions, and just like their name implies, these are watertight. They act as anchors, they hold things together, they're very sturdy. These are incredibly common in your small intestine, so you don't want your partially digested food just leaking in between your cells and your body cavity. So there are these tight junctions, very strong watertight seals that keep your food on one side and your kind of body fluid on the other, so that it only passes through those processing cells and not between them. Other types of connections just act as Velcro to hold cells together, kind of cohesive units, and these are true all over the animal body cavity. Muscle tissue is held together this way. And gap junctions are open channels between cells. These are common in plants but also in animals. So heart cells, to keep the rhythm of the heart consistent and all those cells communicating rapidly with each other, they have lots of gap junctions in heart cells. An interesting thing, and we'll talk more about cancer in chapter 6, but when cells become cancerous, they reduce their number of gap junctions. Basically, they stop talking to the cells around them, which is kind of scary.